So we're going to continue looking at the digestive system, looking at some of the accessory organs that produce hormones here. So the pancreas is the first one we're going to look at. This is going to lie posterior to the greater curvature of the stomach. It's going to secrete its juices into the pancreatic duct and accessory duct, and those will lead to the small intestine. So the pancreatic duct is going to share the common bile duct, and it's going to enter the duodenum at the hepatopancreatic ampulla. When we look at the histology of the pancreas, 99% of the cells are acini, which are going to be exocrine. So the pancreatic juices will be secreted between 1,200 and 1,500 milliliters of them daily. It is mostly water. There'll also be sodium bicarbonate in there to help buffer the acidic stomach chyme. So when things get dumped into the small intestine, they're very acidic. We need to be able to neutralize that a little bit to protect the tissue. So pancreatic amylase will also be included. So proteolytic enzymes, these include trypsin that's secreted as trypsinogen. Chymotrypsin is secreted as chymotrypsinogen. Carboxypeptidase is secreted as procarboxypeptidase, and elastate secreted as proelastates. And then we have pancreatic lipase, ribonuclease, and deoxyribonuclease mixture. That may seem strange, but when you're eating things, you're eating cells, and in order to break down everything, you need to break down the genetic information in there. 1% of the cells are pancreatic islets, the islets of Langerhans. This is the endocrine portion that's going to secrete the hormones. So in this area is where you're going to get your glucagon and insulin, somatostatin, and pancreatic polypeptide from. So this is going to show the regulation here that's going to happen. So you put food in the stomach, that's going to lead to gastrin being produced for your acid production and gastric motility. When the chyme goes into the duodenum, these are going to be the things you end up with. You're going to end up with insulin, pancreatic enzymes and buffers, bile secretion, and dilation of the intestinal capillaries. All of those are going to help with nutrient absorption as it moves into the jejunum, and then that will allow for nutrient utilization by all the tissues. So you're going to get your neural control from the parasympathetic division of the autonomic nervous system, from the vagus nerve. You'll have autoregulation from sensing the presence of fatty acids and amino acids in that chyme. And then hormonal control from the secretion of the enteroendocrine system of the duodenum. So here you can see you've got the parasympathetics. It's going to stimulate the pancreatic enzymes. You're going to have acidic chyme with your partially digested fats and proteins that are going to come in. You're going to have the CCK and secretin that's going to go into the bloodstream, and this is going to stimulate the secretion of the pancreatic juice with bicarbonate and other digestive enzymes. So secretin is going to stimulate the secretion of water, and bicarbonate, the CCK or cholecystokinin, stimulates the secretion of the enzymes from the acinar cells, and that's going to trigger the hydrochloric acid, fatty acids, and amino acids, getting things going. So when we look at the coordination of pancreas, liver, gallbladder, and duodenum, here you can see the anatomy of them. So you can see where the liver is here. Here's the gallbladder. Here's the neck of the gallbladder. So you can produce things in the liver and have it go into the gallbladder. So here you can see the pancreatic wing going into the common bile duct. So that shared tube. And then here's a picture of it in the actual anatomy. So yours is a Word document so that you can separate these pictures or enlarge them if you'd like to be able to see them. So the liver, it is the heaviest gland of the body. It's going to be made up of hepatocytes that are your major functional cells of the liver. It has a very wide variety of metabolic, secretory, and endocrine functions. It will secrete bile, which is an excretory product, as well as other digestive secretions. So the liver is critical to be able to survive. You cannot live without a liver. So bile canaliculi are the ducts between the hepatocytes that are going to collect the bile. That bile is going to leave the liver through the bile ducts that are going to come together to form your common hepatic duct. That will fuse with the cystic duct, and these two ducts form the bile duct. Your hepatic sinusoids, they are highly permeable blood capillaries in there that are going to receive oxygenated blood from the hepatic artery, and then the deoxygenated nutrient-rich blood is going to go out through the hepatic portal vein. So there's 3 different ways to organize the units. Looking at the hepatic acinus is the preferred method, and then the hepatocytes arranged in 3 zones around a short axis with no hepatic boundaries. So functions of the liver. It's going to produce bile, 600 to 1,000 milliliters a day. So the bile salts is bilirubin. It's going to be important for cholesterol, fats, the fat-soluble hormones, and lecithin. It's going to help to neutralize and dilute the stomach acid with its secretions. The bile salts emulsify fats, so most are going to be reabsorbed in the ileum. If you're trying to imagine what emulsifying fats looks like, it allows it to be able to combine into liquids. Soap acts as an emulsification agent. If you go to wash a greasy pan, you'll notice that it's a lot easier to get it clean. If you use soap in there, it allows you to get the fat off easier. That's essentially what bile is going to do with the fats that you eat. It's going to have a storage role for glycogen, fat, vitamins, copper, and iron. So your hepatic portal blood is going to go to the liver from the small intestine. So remember, with portal circulation, it's going to make a second stop. The blood will go to the digestive system. It'll go into the small intestine, and then it's going to make a second stop at the liver. It's important for nutrient interconversion. So it can help turn some of the amino acids into energy-producing compounds when you need to. It will play a role in the hydroxylation of vitamin D, which can then travel to the kidneys where it's hydroxylated again into its active form. So that's important. Vitamin D needs those 2 steps to become active. Plays a big role in detoxification. The hepatocytes remove ammonia and convert it into urea. So we need to process that nitrogen waste. Doing phagocytosis, the Kupffer cells are going to phagocytize worn out and dying red and white blood cells and some bacteria. And then it's got synthesis functions, making albumins, fibrinogen, globulins, heparin, and clotting factors. So if the liver is not healthy, there is a wide range of places that this can show up in the body as not functioning properly. So when we look at the microscopic anatomy, you've got these hexagonal shaped liver lobules, and they're the structural and functional units of the liver. So they're going to have hepatocyte cells that are going to radiate outward from a central vein. The portal triads are going to be found at each of the 6 corners of the liver lobules. And those triads are going to have a bile duct that's going to receive bile from the bile canaliculi, a hepatic artery supply that's going to bring oxygen-rich blood to the liver cells, a hepatic portal vein connection that's going to carry the venous blood with nutrients from the digestive viscera, and then your Kupffer cells with your hepatic macrophages that are found in the sinusoids. So the liver blood supply is going to have 2 sources. Your hepatic artery is going to bring oxygenated blood from the aorta. This is to feed the liver. The hepatic portal vein is going to bring in deoxygenated blood. This is going to have your absorbed nutrients and toxins from the stomach and intestines and hormones from the pancreas. So that's not about feeding the liver, it's bringing stuff to the liver for it to work on. So your hepatic portal circulation is going to take blood from the gastrointestinal tract to the liver before it releases it into the bloodstream. This is important so that the liver can process, absorb, and store the nutrients first. So you'll have the breakdown products of red blood cells from the spleen. The blood gets mixed in the sinusoids. Those hepatocytes modify and exchange the molecules in the blood. So those sinusoids are going to be patrolled by your Kupffer cells. So they are monocyte-derived stellate reticuloendothelial cells. It can do phagocytosis of the damaged red blood cells, bacteria, and other microbes, some of the other foreign molecules and toxins, and then it can store or break down the toxic materials. So this is going to show you've got the two sources of blood that are going to come into the liver. Go to the liver sinusoids, to the central vein, the hepatic vein, and then back to the heart to the inferior vena cava. So this is showing some of those liver sinusoids here, the shapes of them. So here's one that is enlarged a little bit. You can see we've got some of those Kupffer cells in there. This is where you can have bile coming in, portal arterioles and venules, and then going into the central vein. And then these are some actual pictures from underneath the microscope. And then this compares the different units of the liver structure and function here with the hepatic lobule, portal lobule, and hepatic acinus. So this just shows blood vessels here that all are going to go from places in the digestive system and connect to the liver. So when we look at bile secretion, bile is going to come from the hepatocytes and enter the bile capillaries or the canaliculi. Those will empty into the bile ducts. So the hepatic ducts join the cystic duct from the gallbladder to form your common bile duct. The job of the gallbladder is to store the bile. So the common bile duct will meet with the pancreatic duct at the hepatopancreatic ampulla, or the ampulla of Vater. So here you can see your right and left hepatic ducts. Here is the gallbladder. Here is going to be the pancreas. And this is going to join in common bile duct here. And here it comes in at your hepatopancreatic ampulla, or ampulla of Vater. So bile secretion goes from the bile capillaries to the hepatic ducts to form the hepatic duct, the cystic duct from the gallbladder, and the common hepatic duct form your common bile duct to empty into the duodenum. So this is just showing here again where you've got parasympathetic impulses, the vagus nerve in here stimulating the bile production from the liver. Fatty acids in the chyme stimulate the CCK secretion into the blood. That's going to cause contraction of the gallbladder. And then the secretin enhances the flow of the bile. And then you've got your gallbladder here. So we produce about 1 quart of bile a day in the liver. It's a yellowish green color. pH is about 7.6 to 8.6. It's going to have water and cholesterol in it. The bile salts are essentially sodium and potassium salts of the bile acids. The pigment in there is going to come from bilirubin that is broken down from your hemoglobin molecule. The globin is the reusable protein portion that you can use for other things. The heme is broken down into iron and bilirubin. So the gallbladder is going to store the bile. It's going to be a thin-walled, greenish color muscular sac on the ventral surface of the liver. You can live without a gallbladder. Occasionally they will get stones in them that can block things off and become problematic for a person. Without a gallbladder, digestion will be somewhat impaired. A person's still going to be able to live, but they may notice some differences with their digestion as a result of not having one. So the gallbladder is lined with mucosa that fold into rugae, similar to what you have in the stomach. You've got the inner muscularis and the outer serosa. So the bile is constantly going to be delivered from the liver, but the gallbladder will allow it to be stored and concentrated. So it will absorb the water and ions so that it can concentrate the bile up to 10 times. If you don't have a gallbladder, all of that is going to get dumped in and it won't get concentrated before going into the small intestine. So the bile is going to exit through the cystic duct into the common bile duct. CCK or cholecystokinin is what's going to stimulate the gallbladder to release that stored bile. This will largely happen from eating a fatty meal and some proteins. So the hepatocytes will secrete 800 milliliters per day. It's mostly water, bile salts, cholesterol, lecithin, bile pigments, and some ions. Bilirubin is the main pigment. So you get that from your recycled red blood cells. The breakdown product of stercobilin in there is actually what gives feces its brown color. So looking at the color of the feces can actually sometimes indicate if there is something going on with the liver in this breakdown process. So the bile salts are going to do emulsification and helping to absorb those lipids. So gallstones are one of the more common things that can happen. It's precipitated cholesterol and it can block the cystic duct. So when this is trying to push things out and this duct is blocked, that's going to be problematic and it can be quite painful for a person. So this is showing some bile stones in here. You can see the kind of greenish color. So in this illustration, it shows the gallbladder being bright green. It's more of, it typically reminds me more of the color of canned olives, canned green olives, not this bright Christmas tree green that you see in this illustration here. And so we're going to stop there and we will finish up the rest of the digestive system in another video.